ఆఫ్లైన్ అయితే వచ్చిన 10 మంది అని మంచిగా సార్ ఆన్లైన్ ఇబ్బంది.
Hello. Hello. Please respond, Lalita. Yes, this is Amma. So today we are going to discuss about fungal taxonomy. So before going that one, so what is the meaning of fungal taxonomy? Okay. So the fungal taxonomy, actually this is of the vast number of fungi inhabitate the earth. Henceforth compiled the fungal species members with those other groups of several geographical regions that had been explored extensively for the biodiversity. He put conservative figures 1.5 million species of fungi worldwide, which only about 79,000 species we are looking worldwide fungus. Yes, so next now you see here, what is the main criteria? What is the main criteria classifications? Okay, yes, so now you see here, the mycologist they use many types of characters. The basis of for their classifications. Here the character is any attribute in the feature of an organisms that can serve as the basis of comprehensive with other organisms. The different expenses of the characters are known as character stats. For example, the fungal spores in group of under study may be smooth or ornamented. The characters may be borrowed from morphology and anatomy, ultrastructural features. The physiological, biochemistry, and other nucleic acid sequencing of various other characters from the basis of building, that is phylogenetic trees, and all types of can and should be used to phylogenetic analysis. The first, the criteria in that one criteria, to see mainly we are taking first morphological characters. The gross morphological features are easily discernible, low magnifications. Example of these characters include the basic shapes of the fungal thallus and from the color and size of spores producing structures. By 1887, the basis of fungal life cycles and morphology were established and used for classification. Gross morphological features served to establish a higher level taxa, such as those within Ascomycotina and Mycorrhizae. The gross morphology also may be used in group such as Ascomycotina, wood decaying member of Mycorrhizae. So this is the first one. The first one that is in morphology. Yes. So next now you see anatomical characters. Anatomical character means the fungi can be observed with the compound microscope. Crash mounts, that is of hind port sections, that is next one, or other one that is of microtome sections. So that is embedded materials. Some of the anatomical characters used in mycology include the arrangement of hyphae comprising the tissues of spore producing structures, the arrangement of ASCI, mean that is of in Ascomycotina and Basidia sterile structures within the spores producing structures of analysis hyphae structures. Mean this is the second one. The second one, that is in anatomical structures. So the anatomical structures means when we are looking what type of structures in there in anatomy. So what type of spores is there, and what type of development we are looking at anatomical structures. So based on that, so when you are doing the classifications, we call that is in anatomical structures. So next, now you see another one, that is ultra structure of features. Okay, ultra structure of features. The host of new character become available from the development of electron microscope and even empowering techniques in specimens preparations, although the character derived from ultra structures. Okay, so these technique used by extensively in the resolve minute spores, that is of an ornamentation, and in some cases details of contain, that is of an ontogamy. So this is ultra structural features. What type of cell structures you are looking in that one? So based on that one, we are going to classifications. We call that is in ultra structure features. Yes. So next one, that is of in physiological structures. The physiological structures used in classifying fungi include decay type, especially in wood decaying species. Of example, that is in Mycology and other post-relation pathogenic fungi. Both these characters and indicate of complex physiological process. So this is the physiological process, means some of the fungi we are looking, for example, that is a Mycology, and Mycology we are looking above the plant. So what is the relationship between plant and Mycology? So based on that one, we are going to the classifications. We call that is in fungal characters. And now you see the next one, that is in biochemical characters. Sir, different type of the fungals are produced, different type of the chemicals. So based on chemicals, so we are going to the classifications. So here now you see, for example, biochemical means in aspergillus, yeah. So it is produced to some chemicals. So these are all the fungus we are using to different type of the chemical industries, different type of the medicine industries. So based on what type of chemical it is produced. The number of biochemical characters have been shown an important chemical techniques such as that is in chromatography, protein electrophoresis have been useful in, that is so that is we are all using these type of the chemicals. Yeah. So next one, that is what type of fungal pigments, isosomes. So chromatography. So these different type of the chemicals are produced to different type of the fungi. So based on that chemicals we are going to the classified. We call that is in so that is biochemical characters. And next one you see reproductive characters. So reproductive characters mean this is also most important. Some of the fungi is produced to conidias. Some of the fungi is produced to zoospores and aplonospores. Okay. So what type of sexual reproduction systems? Different type of the fungi, different type of the sexual reproductive organs are terminated or produced. Based on that one, we are doing the classification. For example, you see the fungi reproduce both, that is sexually and asexually. Means asexual means here we are looking conidiophores and aplonospores and zoospores. Okay. So next now you see it in sexual reproduction systems. So in sexual reproduction system means, so one is a plus, second one that is a minus. So here we are looking the plants are produce a different type of the so sexual reproductive organ. Structurally, lot of variation one sexual part to other sexual part. So based on that one, we are doing the classifications. So here now you see that is the fungi classified in the base of sexual characters because they are considered to be stable. These characters are not useful at the major taxa level, but also even the species and still at a lower level. So based on that one, we are going to classifications. We call that is in reproductive characters. And next, now we see here that is molecular levels. Molecular levels means atomic structures. So based on that, based on the atomic structures, so we are going to the classifications. We call that is in molecular methods classification. For example, now you see here the traditional methods identifications depend on including of fungi, depending on including the fungi, and let identify them based on their morphological features. So molecular method levels in genetic diversity in the fungal produces, although the molecular methods have been used to study of bacterial communities. Very little research have been undertaken for fungi. So, for example, that is in PCR and RAPD and RFLP. So, next one that is polymerized chain reactions. Next one that is in polymerized chain reactions. So, based on the PCR method, PCR that is targeting these 16S or DNA has been used extensively to study prokaryotic diversity identifications and for the production of phylogenetic relationship. Yeah. So polymer chain reactions, what type of DNA we are looking at that one. Okay. So based on that one, based on the DNA, based on what type of structures we are looking in that one. So we are going to classification that is in PCR. Next one that is of in random amplified polymorph DNA RAPD. This is useful tool to diagnosis, isolate for population of individual species and possible to distinguish among the species. It is basically PCR assisted technique. It indicates the DNA polymerized is a different species and isolate of individual species. So this is a DNA. So and next now we see here RFLP, restriction fragment length polymorphisms. Okay. So this is another. This is also one of the molecular based fungal taxonomy. Okay. So molecular based technique aimed to reveal in DNA polymorphism. DNA based on the DNA structures we are going to classify it. That is often. So next one that is often protoplast and nuclear fusion. Protoplast and nuclear fusions. Yes. So next now you see here. So this is one of the most important types of classifications. So generally, the classifications. What is the meaning of in classifications? Mean identifications. The classification are generally 3 steps is there. So one it is in identifications. Second one that is of in nomenclature, and so third one that is in classifications. The first one that is in identifications. So where it is available, so where we are looking, how it is observing, that is identifications. And second point, second one that is in nomenclatures. Nomenclature means so based on the area, so based on the scientist, different type of the sections you are looking in that one. That is of nomenclatures. And third one, classification. This is most important. So these classifications are generally 3 types. One it is in artificial classifications, second is the natural classifications, and third one that is in phylogenetic classifications. So artificial classification means so based on the some we are the scientist giving some characters. So this is this much of height is there, this type of color is there. So we are looking we are giving the some characters based on that characters. So we are going to classification, we call that is in artificial classifications. So and next one that is in natural classifications. Natural classification means we are only what type of structures you are looking, what type of structure you are observing in that one. Means here we are not giving any type of characters. Whatever we are looking, whatever we are observing, based on that one we are doing the classifications. We call that is a natural classifications. And next last one that is in phylogenetic classifications. So already you know very well phylogenetic classification means so where it is coming, how it is coming, so which one is ancestors, so what type of the characters we are looking in that one. So based on the ancestors, so we are going to the classifications. We call that is in phylogenetic classifications. Okay, So this is fungal criteria and types of classifications. Yes. So now you see the next entire the fungus are divided mainly two classes. Entire the fungus are divided mainly two classes. Okay. So number one, that is in Myxomycota. Number two, second one, that is in Eumycota. First one, Myxomycota. What is the meaning of Myxomycota? Plasmodium or pseudoplasmodium are present. That is in Myxomycota. Plasmodium, pseudoplasmodium are absent. That is in Eumycota. Okay. Assimilative phase, typically filamentous plants. These are all in Eumycota. Okay. Yes. Again, now these Myxomycota identification characters, assimilative phase, a plasmodium. Okay. So next one, that is in assimilative phase, free living amoeboid which unite as pseudoplasmodium before reproduction. So all together we call that is in Myxomycota. So next, so another one that is most important here, Eumycota. That is in Eumycota. Okay. So in Eumycota, based on the fungal structures, these Eumycota are divided in 5 categories. One, it is in Masticomycotina, Zygomycotina, Ascomycotina, Basidiomycotina, and Deuteromycotina. Okay. First point, what is the Masticomycotina? What is in Masticomycotina? Yes. So now you see here in Masticomycotina, most important point is that is in spores, motile cells, that is zoospores present. Okay. Perfect state spores, typically oospores, because that is in Masticomycotina. So next one, now you see here, zoospores posteriorly uniflagellate, flagella whiplash type. Zoospores not posteriorly uniflagellate. Zoospores anteriorly uniflagellate. Flagella is a tin cell type. Zoospores biflagellate, posterior flagellate, whiplash type of, anterior type of. So all these are all the characters. These are all the characters. We call that is in Masticomycotina. So next point. What is the next point? That is in second one, Zygomycotina. Vegetative phase, hyphae or non-septate. Mycelium or cenocytic. Asexual reproduction by means of non-motile aplanospores. That is in Sporangiospores. The sexual reproduction by gametangial copulation that result in the formation of zygospore. Okay. So these are all the main characters of Zygomycotina. Yes. So next, now you see what type of keynotes of in Zygomycotina. Saprophytic generally. In parasitic or predaceous, their mycelium is present immersed of host tissues. That is in zygomycetes. Parasitic in the birds of arthropods. Okay. So this is of in millipedes insect larvae. So these are all the characters of in Zygomycotina. One, it is in Ascomycotina. Second one, that is in Zygomycotina. So Masticomycotina and Zygomycotina. Next, third one is most important, Ascomycotina. Here now you see the first one is Masticomycotina, second one is Zygomycotina. Masticomycotina, it is produced to juice spores. Okay, second one. So Zygomycotina, it is produced to aplanospores. Juice spores, Masticomycotina. Aplanospores are Zygomycotina. Third point, that is in Ascomycotina. Ascomycotina. This is third group of Eumycota division. In Ascomycotina, always it is produced to ascospores. Okay, you see here, how can we identify in Ascomycotina group? Ascomycotina, when it is completed copulation, when it coming together, when it is contact, peer formation, one fruiting like body, fruiting bodies, okay, ascospores, ascogenous hyphae, like Thales mycelium or yeast like, okay, so that is a Ascomycotina. Yeah. See, after the reproduction system, these Ascomycotina plants or formation one fruiting like bodies. Okay, so based on the fruiting like bodies, we are saying that is in Ascomycotina. Ascoscarp or ascogenous hyphae present in Thales mycelium. This is the first characters. Okay, Asci, that is of bitunicate ascocarp, or that is of in ascostroma. Asci typically unitunicate and binucleate ascocarp of apothecium. Apothecium means this is one type of fruiting bodies. Okay, so these are all in Ascomycotina group. These are all in Ascomycotina group. And see now some more points of in Ascomycotina, that is of in well-developed mycelium. Well-developed mycelium with septate hyphae. First point. Second point, asexual reproduction structures, that is in chlamydospores and conidias, or other one that is in sclerotia. Okay, so next now you see the sexual reproduction result in the formation of sac polled ascus. Ascus contains ascospores. Fruiting body is generally known as ascocarp. So because this is in Ascomycotina, and these Ascomycotina, so classes is there. Okay, Ascocarp, ascogenous hyphae like Thales mycelium or yeast cell. This are in Hemi-Ascomycotina, and Ascoscarp or ascogenous hyphae present Thales mycelial, that is of in Mycoascomycotina. Okay, so these are all in Ascomycotina group. After the Ascomycotina, so next one that is in Basidiomycotina. Okay, Basidiomycotina. This is of in second, third, fourth one. That is of in Basidiomycotina. Means these Basidiomycotina, it is produced to basidiocarp. Okay. So here also we are not looking into true sexual reproduction systems. When the life cycle is completed, the spores are comes to outside the which one that is of sporangium or spores chambers. Only spores. There is no sexual reproduction systems. Spores are comes to outside the chamber. That spores are goes to where the host. When entered in the host, okay, so on that area it is modified as a plus or minus. But these type of the section we are looking, that is in Basidiomycotina. Okay, so we call that is in basidiocarp. We call that is in basidiocarp. So next one that is in Deuteromycotina. Next one that is in Deuteromycotina. So in Deuteromycotina, the sexual reproduction system is totally absent. The sexual reproduction system is totally absent. There is no any sexual reproduction systems. There we are looking in only asexual reproduction systems. Okay, so this is all the 5 types is there in fungal. Okay, so classifications. One it is in Myxomycota, and second one that is in Geomycota. Yeah. So after this one, so next now you see, so we are going to the third one. So here what is the third one that is in mycorrhiza. Okay. Yes. So now you see what is the meaning of mycorrhiza. Yeah. So mycorrhiza means myco fungus. Okay. That is in rhiza. The some rhizoids. Rhiza means rhizoids. Some fungus we are looking in only where the root that is in mycorrhiza. Just 1 minute. Here now you see the term mycorrhiza was coined by German botanist. His name is Frank in 1985. Okay, so myco means fungal, rhiza means root. The mycorrhiza composite organs of root for fungus are formed is most species of angiosperms and gymnosperms and some pteridophytic plants. Some of the fungus we are looking in only where the root systems, that is gymnosperms or angiosperms or pteridophytic plants. When the fungus you are looking from the root systems, we call that is in mycorrhiza. So generally, so these mycorrhiza are 7 types is there, but one mostly 2 types, mostly 2 general, that is of 7 type also we are looking. First one that is in ectomycorrhiza, second one that is in endomycorrhiza. First one that is in ectomycorrhiza, second one that is in endomycorrhiza. Okay, so here now you see the ectomycorrhiza, the most common type of brown ectotropic mycorrhiza with a thin mantle and that is a hot-tick net. The mantle may even be almost absent. This is an ectomycorrhiza. Ecto, endo, outside. Outside means, for example, this is a cell. Above the cell, we are looking that is of an ectomycorrhiza. Inside the cell, when the development of fungi, we call that is in ectomycorrhiza. So endomycorrhiza and ectomycorrhiza. So next, and general 7 types is there. So that is of VAM. VAM means vesicular orbuscular mycorrhiza. So this is endomycorrhiza. And some third group of ecto and endomycorrhiza means some fungi we are looking inside and outside. That is of ecto endomycorrhiza. And third one that is of in orbital mycorrhiza and monotropid mycorrhiza and ericoid mycorrhiza and the fungi we are looking in orchids, we call that is of in orchidaceous mycorrhiza. So this is of in mycorrhiza. Mycorrhiza. So and so next to mushroom cultivations. Mushrooms. So I think already you know very well mushrooms. So what is the meaning of mushrooms? Mushrooms are fruiting body. Mushrooms are fruiting body of fungi, especially Ascomycotina and Mycorrhizae. The term mushrooms is generally used for edible agaricus or mushrooms. The other term that is of in toadstool, which is used to non-edible and poisons mushrooms. Non-edible and poisons mushrooms. Okay, so what is the structure of mushrooms? See, so this means almost all. See, there are more than 2,000 species edible fungi all over the world wise, almost all. So mushrooms means here it is totally fungus. So these fungus are also used into the edible fungus. So now you see here. Hello, please respond.